Hello, I am Professor Sims, and this video is about enzymes, cellular respiration, and fermentation. This is the 7th of 10 lessons included in my Fundamentals of Microbiology course. If you are currently enrolled in this course, please consult the syllabus and Moodle site for assignments and other course information. So lesson 7 is we're looking at metabolisms, autotrophs, heterotrophs. We'll be looking at oxidation reduction reactions, ATP, FAD, NAD+, NADP+, structural components of enzymes, electron transport systems in prokaryotes and eukaryotes, substrate level oxidative phosphorylation, chemiosmosis and proton motive force, and then aerobic, anaerobic respiration, and then fermentation, including end products and how it relates to aerobic and anaerobic respiration as well. So the first thing, I'm going to talk about metabolism in general. Metabolisms where you have breaking down of complex molecules. So if you have a large molecule, say like a starch, and you break it down into its monosaccharides or sugars, then that would be a catabolic reaction, catabolism. If you are building up complex molecules from simple molecules, like when amino acids are being built up into a protein, for example, that is anabolic reaction, anabolism. Breaking down is catabolic, catabolism. Building up is anabolic, anabolism. Organisms are classified according to their sources of carbon. Remember, carbon is the molecule of life. Autotrophs can convert an inorganic carbon dioxide into an organic carbon molecule. Heterotrophs use an organic carbon compound, so they can't convert the inorganic to organic. They have to have an organic compound. Autotrophs can take their energy from CO2. You see this in plants. And then heterotrophs, they have to eat other life forms. Organisms also can be classified according to their energy source. So it is a different thing. It's not, because carbon is more than just energy. Like, you can derive energy from carbon, but it also has structural things and other. But specifically energy source, we're talking about phototrophs, chemotrophs, organotrophs, or lithotrophs. So phototrophs can get their energy from light. Chemotrophs need chemical compounds to make energy. Organotrophs use organic molecules, so ones that have carbon, organic carbon, and lithotrophs can use inorganic chemicals. So this is figure 8.2, and this is showing the process of metabolism, and it is showing the difference between catabolism and anabolism. The anabolic pathways require energy, and they synthesize larger molecules. Catabolic pathways actually release energy by breaking apart larger molecules into smaller ones. An electron carrier accepts electrons and then serves as an electron donor in a redox reaction. So a redox reaction essentially is movement of electrons or hydrogen ions back and forth across a cell membrane or mitochondrial membrane. So your electron carriers are the things that accept the electron and then later donate the electron in these redox reactions. FAD, FADH2, NAD+, NADH, NADP+, NADPH. These are all examples of electron carriers, and they are really important in metabolism. And you'll see why over the course of this lesson and the next one. ATP is short for adenosine triphosphate, and this is the currency. This is energy for cells. And what it means when it says the currency, the energy currency, is it's the way that cells can use energy. It's this molecule. It stores energy in its phosphate bonds that can be used in reactions later on. Enzymes are catalysts. Catalysts lower the activation energy that's required. So this is the energy that's required to activate a reaction to make something form products. Exergonic reactions are ones that release energy, and they can proceed without enzymes, but they're usually at a very slow rate. Endergonic reactions require energy beyond that of the regular activation energy, so they absolutely do need enzymes. In cells, endergonic reactions are usually coupled to exergonic reactions, which can also help reduce the activation energy that's needed. So exergonic releases, endergonic requires. So let's look at this again. Anabolism, is that exergonic or endergonic? Endergonic. It requires energy. Catabolism is exergonic because it releases energy. This is figure 8.3, and it's showing the energy that's released by dephosphorylation of ATP. And essentially all that's happening is you got adenosine triphosphate, so named for its 3 phosphates. And when it's dephosphorylated, it loses one of its phosphate groups. And then when that happens, there is energy that is released. In order to regenerate ATP, you have to go through a process called phosphorylation. It's very broad schematic, but it's solar or chemical energy is added to ADP, and it's phosphorylated to form ATP, and then when that phosphate group is cleaved, it releases energy for metabolism, and then that cycle continues. This is 8.4, showing an exergonic reaction coupled to an endergonic reaction. This is coupling ATP phosphorylation to catabolism. And then ATP dephosphorylation is then coupled to polypeptide formation, which is an example of anabolism. And again, you see this cycle. We are going to talk a lot more about glycolysis and Krebs cycle in lesson 8. We will go over oxidative phosphorylation just a little bit later in this lesson. Most metabolic processes need enzymes, and it's because they need the reactions to occur fast enough to sustain life. In other words, there's some reactions that could take place without enzymes, but they would be so slow that the organism would not be able to get the energy that it needs. It would die. Lowering activation energy increases reaction rate. It makes it faster. Enzymes are not, this is important, enzymes are not consumed. They are not altered. They're not converted in reaction. They remain intact. The molecules that are occurring at the beginning of an enzymatic reaction are called substrate, and then the enzyme converts substrates into what are known as products. Substrates bind to an enzyme's active site, and the process alters the structure of the active site and the substrate. This is called the induced fit model. This is actually a modification that came from an earlier model called the lock and key model. The lock and key model said that the substrates bind to the active sites because they have a certain stereochemistry and only certain substrates can bind to only certain active sites. The induced fit model is very similar to that, except that it does say that the active site can alter its shape in order to fit the right substrate. And the substrate can also alter its shape in order to fit and target active site. This is figure 8.5, showing how enzymes lowering the activation energy of a chemical reaction. So if the green line here is the energy required for a reaction without an enzyme, then the red line here is the energy required with an enzyme. So it goes a lot faster with an enzyme. Figure 8.6 is showing how the induced fit model works. So we're looking at enzymes, substrates, active sites, products, all of this jargon. This figure shows very nicely what everything is. This here, this little gray looking, weird looking thing, is the enzyme. Substrate. So a substrate is one substance that is going to be altered or converted into something else. So the substrate is going to be converted into a product. So if you had hydrogen and hydroxyl group and those combined to form water, then hydrogen and hydroxide would be the substrates. The product would be water. This is showing substrates that are being bound to an enzyme, and they are specifically bound to the active site of the enzyme. And the induced fit model, unlike the lock and key model, it says that the enzyme does slightly change shape as the substrate binds. When then the substrate is bound, the chemical reactions occur. This is the part where the enzyme has reduced the activation energy needed for the reaction to occur. Once the reaction is finished, the products are released from the active site of the enzyme. Cofactors and coenzymes, apoenzymes and holoenzymes, and inhibition. Cofactors are inorganic. They're inorganic ions that stabilize enzyme conformation and function. So cofactors, they're like little helpers for the enzymes, and they are inorganic ions. Coenzymes are organic molecules that are required for proper enzyme function, and they're often derived from vitamins. The two main differences here are cofactors are inorganic ions, so they're also pretty small. Coenzymes are organic molecules, so they're bigger. And they both are involved in stabilizing and helping enzymes do their job. An enzyme that doesn't have a cofactor or a coenzyme is an apoenzyme. An enzyme with a cofactor or a coenzyme is called a holoenzyme. So if it doesn't have it, it's APO, right? If it does have it, it's holo. And then you have competitive inhibitors and non-competitive inhibitors. And you also have what is known as feedback loop, feedback inhibition. Competitive inhibitors bind to an enzyme's active site, and they get in the way. They prevent substrates from binding because the active site isn't available for the substrate to bind to. Non-competitive inhibitors, they're also known as allosteric inhibitors. They induce a conformational change in the enzyme that prevents it from functioning. What that is a conformational change means that its structure changes, its shape changes. Always remember that when you're talking about enzymes and when you're talking about proteins, because enzymes are made from proteins, that structure equals function. So if you've got something that's making an enzyme change its structure, then that enzyme won't be able to function. And then feedback inhibition, this is when the product of a metabolic pathway, so a metabolic pathway is where there are several steps involved, so several reactions that happen in succession, and we're going to see that when we look at glycolysis and Krebs cycle. So during a metabolic pathway, a product is formed early on and that binds to an enzyme, and it prevents the synthesis of a product later in the pathway. So it gums up the works. It's kind of like throwing a monkey wrench in the spoke of a bike or something. So figure 8.7 shows an apoenzyme that becomes active by binding of a cofactor ion or a coenzyme molecule. This gray guy right here, all by himself, is an apoenzyme. He doesn't have the cofactor, he doesn't have a coenzyme. Once a cofactor or a coenzyme binds to this apoenzyme, then that becomes a holoenzyme, and then a substrate can bind to it. Just remember, apo means it doesn't have cofactors or coenzymes. Holo means that it does. Enzyme activity is regulated by either competitive or non-competitive inhibitors. So this is figure 8.8. A competitive inhibitor here, you see that it's added to the active site of the enzyme. And the substrate is coming along to add, and it can't because this guy's in the way. So this is some kind of molecule something has added to the enzyme, and the substrate that's supposed to add there can't because it's in the way. So that's competitive inhibition. Non-competitive inhibitors, they're not bound at the active site. They bind at what is known as an allosteric site. It's kind of a secondary site. But when they bind there, it changes the shape of the active site. So the substrate can no longer bind to the active site. So this is allosteric inhibition, which we just saw with the non-competitive inhibition. You have this molecule that adds to an allosteric site. It changes the shape of the active site, and the substrate can't bind anymore because it doesn't fit anymore. Allosteric activation actually kind of does exactly the opposite thing. you have an enzyme that already has an altered active site, probably because some other inhibitor bound to it first, and then an activator adds, and it alters the active site. It puts it back to where it's supposed to be. So it does go both ways, depending on what molecules you're dealing with, what enzymes you're dealing with, what step of a pathway you are dealing with. This is a feedback inhibition loop. So the end product of the pathway, well, it turns out this end product becomes an allosteric inhibitor, which can prevent the addition of this substrate to this enzyme. So if this is enzyme 1, remember that enzymes are not used up in reactions. So as long as you have these 3 enzymes and you have the substrates, this substrate floating around in the cytosol or wherever, ideally this pathway could just go on and on and on and on and on forever, because once this reaction happens, this enzyme doesn't get used up. It's still there. You have the substrate adds to this enzyme, it makes this product. This becomes a substrate for another enzyme, makes this product. This becomes a substrate for another enzyme, makes this product. And then this product actually adds to the first enzyme and prevents the pathway from repeating. And that's why they call it feedback inhibition, because it's a product that is a product of reactions that already occurred in a pathway, and it prevents the pathway from repeating. Most ATP is generated during what is known as oxidative phosphorylation, electron transport system. Okay, ETS, and you also have heard ETC, electron transport chain. These are talking about the same things. It's just that this book calls it electron transport system rather than electron transport chain, which is kind of unusual. But it's a series of membrane protein complexes and electron carriers. It's embedded in the cytoplasmic membrane in prokaryotes, and it's in the mitochondrial membrane in eukaryotes. See, because prokaryotes don't have mitochondria and eukaryotes do, and the mitochondria and eukaryotes are primarily responsible for making energy. Each electron transport complex has a different redox potential. Redox potential is the tendency of a chemical to either accept electrons and be reduced, or donate electrons and be oxidized. So redox is short for reduction oxidation. It's reduced if it accepts electrons. It is oxidized if it releases or donates electrons. Electrons will move from electron carriers that have more a negative redox potential to those that have a more positive redox potential. You know, I think I'm going to pull up a video to show y'all that kind of helps me explain this. But in aerobic respiration, the final electron acceptor is oxygen. In anaerobic respiration, the final electron acceptor has to be an alternative to oxygen, and there are many: nitrogen, sulfur, all kinds. As electrons are passed from NADH and FADH2 through the electron transport chain, at first you lose energy, but that energy is actually stored by the pumping of a hydrogen ion across the membrane. What is happening is energy is harnessed by allowing the hydrogen ions to diffuse back through the membrane through a chemiosmosis, and then that is performed using the enzyme called ATP synthase. And as the ions diffuse down their electrochemical gradient, the ATP synthase makes ATP from ADP, and this process is called oxidative phosphorylation. So it's making adenosine triphosphate from adenosine diphosphate. In aerobic respiration, this process, oxidative phosphorylation, forms approximately 34 ATP molecules in the electron transport chain. In anaerobic respiration, because they have to use an alternative electron acceptor, it doesn't form nearly as much ATP. In fact, it can form a very widely variant amount of ATP depending on what its alternative electron acceptor is in the chain. So it can be 1 ATP to generally a lot of what you're going to see is going to be between 2 and 4, but that's because they can't use oxygen as their electron acceptor. I'm going to pause the presentation right here, and I'm actually going to show you a quick video on the electron transport system and ATP synthesis.
The energy stored in NADH is used to generate a proton gradient across the mitochondrial membrane, and the energy of the proton gradient is used to make ATP. When glucose is oxidized during glycolysis and the Krebs cycle, the coenzymes NAD and FAD are reduced to NADH and FADH2. Inside the mitochondrial matrix, the electrons from NADH are transferred to the electron carrier coenzyme Q by NADH dehydrogenase, and the protons are transferred across the membrane to the intermembrane space. Coenzyme Q carries the electrons to the cytochrome BC1 complex. As the electrons move from the BC1 to cytochrome C, more protons are carried from the inside to the outside of the membrane. Electrons are also transferred from FADH2 to coenzyme Q, with the protons being transferred across the membrane. Cytochrome C transfers electrons to the cytochrome C oxidase complex. Protons are also transferred to the outside of the membrane by the cytochrome C oxidase complex. The cytochrome oxidase complex then transfers electrons from cytochrome C to oxygen, the terminal electron acceptor, and water is formed as the product. The transfer of protons to the intermembrane space generates a proton motive force across the inner membrane of the mitochondrion. Since membranes are impermeable to ions, the protons that re-enter the matrix pass through special proton channel proteins called ATP synthase. The energy derived from the movement of these protons is used to synthesize ATP from ADP and phosphate. Formation of ATP by this mechanism is referred to as oxidative phosphorylation.
So let's have a look at figure 815. This is looking at the ATP synthase. And like you saw in the video, this is an integral membrane protein through which the hydrogen ions flow down their electrochemical gradient. And this results in the formation of ATP from ADP in a phosphate group. And this specific part of the electron transport chain, it is a result of proton motive force, and it is known as oxidative phosphorylation. Also in the video you saw the cytochrome C complex. You're going to be learning a lot more about that in the lab. We're going to be testing for oxidation via cytochrome C oxidase, so be looking forward to that in lab. Fermentation, we're also going to do some fermentation experiments in the lab this week. Fermentation uses organic molecules as the final electron acceptor. Notice it did not say oxygen. Keep that in mind. Fermentation does not involve an electron transport system, so this is an entirely different metabolic pathway. No ATP is made directly by fermentation. What happens is fermentation results in NADH production, and then NADH is used in glycolysis as an electron acceptor. And we're going to talk about glycolysis in lesson 8. Fermenters make very little ATP. Fermentation, the process itself, does not make ATP. It makes NADH. But that NADH is used in glycolysis to make ATP. But it's still a very small amount. Okay, so it doesn't make nearly as much as would an electron transport system. Microbial fermentation, it's often used to produce food like sauerkraut, sour cream, lots of different things. They use it in production of pharmaceuticals, and also, and we're going to see this in lab, it is used in processes of identifying unknown microorganisms. In lactic acid fermentation, you have the pyruvate accepts electrons from NADH, and then that produces lactic acid. Homolactic fermentation produces only lactic acid as the fermentation product. Heterolactic fermentation can produce lactic acid, it can produce ethanol, it can produce acetic acid, and it can produce carbon dioxide gas as well. So when we do our fermentation tests in the lab, you'll see the byproducts of fermentation and the products of fermentation, and we'll be able to test for them by measuring pH, because acids and alcohols lower the pH. And we also have a method that we're going to learn a procedure to test for the production of gas as well. Normal microbiota, so these guys that live in your body, remember normal flora, can prevent the growth of pathogens. So because they're producing lactic acid, they're dropping the pH. A lot of bacteria can't live in low pH. And this whole process is really important for your gastrointestinal tract health, your colon health. Ethanol fermentation, pyruvate is decarboxylated. This releases CO2, and the product is acetaldehyde. Then that acetaldehyde accepts electrons from NADH, right? So acetaldehyde is the electron acceptor in ethanol fermentation. Acetaldehyde is then reduced to ethanol. This process is used to make alcoholic beverages, and it also makes bread products rise because of this release of CO2, and it's also used in biofuel production. Other fermentation products such as propionic acid, they can provide distinctive flavors to food products. Then you have acetone, butanol, ethanol fermentation, makes chemical solvents and pharmaceuticals, right? And then microbes can be identified by their substrate or product or byproducts of fermentation. Figure 817 is a chemical reaction of alcohol fermentation. So these are the three steps: pyruvic acid, acetaldehyde, NADH, and then ethanol production. This concludes the Lesson 7 material. I want to thank you guys, as always, for watching. Don't forget to do the reading. Check the description for more videos related to these topics, and leave your questions for me in the comments section below.